All right, it's 0 minutes and 0 hours and 0 days, and we are starting this 24-hour around the globe event on the Human Reference Atlas. It's my true pleasure to welcome you all. We have a great lineup of speakers. I'm going to guide you through it a little bit, and I'm also giving it over to Andy, my co-host.
Hey everybody, I'm Andy. Nice to have everybody here. This is our second 24-hour live stream. If you joined last year, we had a live stream about science mapping. We learned a lot from this first iteration. We made it through all the 24 hours. We went to multiple continents, to multiple speakers. My favorite part last year was a live music session about improvising. It was pretty amazing. I had very fond memories of this. This year we're trying to come back even stronger.
Thank you. All right, let me give you a tour of what's going to happen over the next 24 hours. So I think all of you found this website. And if you go through the events, you also learned how to connect. There's the Zoom link, the magic one that connects you to those 24 hours of content. And we will bring in scholars and practitioners from around the world to present their work in a more general audience-friendly way. Typically, we scholars connect to other scholars and we present very abstract and sometimes very difficult to understand concepts to each other. But this is really an event where we are trying to make it understandable to anyone, to my daughters, to my mom, to anyone who is interested in science and would like to benefit from the fruits of science. We hope this event will kick off the holiday season, and you see our little gnome here also illuminated and happily sitting there. And we hope it also creates ever more excitement for the value of science and gives energy to those which have been impacted by this ongoing pandemic. As you saw, we have a short welcome, Andy and myself, just giving you a brief introduction into this effort that many scholars around the globe are participating in, trying to map the human body at single cell resolution. Then there will be trivia on this topic also, so you can win prizes. So watch out for this. At 12:40, Todd Theriault will take over and guide you through a number of trivia questions. In the meantime, you might have seen that HubMap, an NIH-funded project, is funding infrastructure to have people explore tissue data that now exist for different parts of the human body, many different organs. And you can go to an exploration user interface like this one. You can search for anatomical structures or cell types. And you can also explore this data geospatially to see what kind of tissue data exists where. It's not just HubMap which is working on this. There are many different consortia which are listed here on the left-hand side. And these consortia care about very different organs. So here also listed, and actually this list is growing by the minute. And ultimately what we need is like a map of the world where you have specified and well-defined 2D and 3D areas in the human body, which are named, and it's non-trivial to have experts agree on names for these areas or cell types for that matter. But ultimately, there needs to be agreement around cells and anatomical structures, probably 5,000 or if not more, across consortia. And we are trying to accomplish this by agreeing on data, formats, and standards, such as so-called anatomical structures, cell types, and biomarker tables. And you see here a visual rendering of those, where you have, for instance, here the human body, and then just one organ out of many. And that one organ links over to different anatomical structures that are known to exist inside of the heart. And then, of course, inside of the left ventricle of the heart, there are other anatomical structures. And then you can follow these linkages to specific cell types, which are potentially in different areas of the heart, and where different biomarkers have been identified as important to characterize these cell types. And it's not just gene biomarkers, it's also protein and lipids and metabolite biomarkers that we are now using to understand what cell types exist where and how do they also, in some cases, for instance, for immune cells or blood cells, flow throughout the body. In the 24 hours that we have planned here, we will have a panel on what is a human reference atlas. There are many different efforts, and different efforts might have very different definitions for how to construct a human reference atlas or how to even define it. And so we are very pleased to have a high-powered set of experts here in this panel, which are going to try to answer your questions and also expert questions and presenting their work and their efforts. In the following, we also have a panel on who funds the Atlas. And here we have funders from Europe, but also from the US, here NIH, and private foundations, which are all interested to advancing our understanding of the healthy human body, helping us to understand what happens when a disease comes in and how can we all live a better, more healthy life. Next, we have a session on the creation of Homo perfectus. I'm a deep believer that the more you can go back in time, the more you can also go forward. And this review of what has been done so far in human anatomy work by Marie Dauenheimer, a board-certified medical illustrator, I think is very important for our understanding of how the human body has been mapped, conceptualized, and also ultimately communicated to a larger audience of specialists and general audiences, so that we can learn from that as we go and map the human body at single-cell resolution. Next is presentation on molecular landscapes. Some of you have been seeing the DigiZyme work on, for instance, here, cell signaling technology. It's beautiful, stunning work in animation. You can't miss that particular presentation. Then there is a panel on naming cells using validated antibody panels moderated by Andrea Radke, which brings in quite a bit of industry expertise and also, I think, features walks through one of these labs where some of the tissue data gets generated. So please come in and join us for this panel. Then there is a session on how to name cells. Again, it's not easy to have experts agree on names for cell types and cell states. And it's not just names that we need, but also the outlines in 2D and 3D. So for each cell, we want to understand what is the extent in space, but also what is the semantic name and term for that particular cell. And as you might know, there are 37 trillion cells in our human bodies, and it matters where they are. Space matters. And so here we have two presenters, and some of you might have seen Jen Fang's beautiful animations online of, for instance, spatial transcriptomics. And after this session, you absolutely will know more about what spatial transcriptomics is. Segmenting cells is important in 2D, but ultimately we are interested in hierarchies of cells. And so here, two gentlemen will present on cell segmentation, but also hierarchical cell segmentation. At 8 p.m., we have a panel on human atlas design in multiple dimensions. Todd Seriold is going to be our moderator, and he's going to interview and present work by Heidi Schleelein and Rachel Bayema. Both of them have been working in HubMap on 2D and 3D reference organs, and it's beautiful work and meaningful work and scientifically accurate work, and you get a behind-the-scene look of how the atlas is created. At 9 p.m., there's a panel on human body exhibits. Stephen Utsu is going to be our moderator. He's a long-term collaborator of our team. He comes from the Science Museum world. He just joined the National Museum of Mathematics, and he invited an amazing set of experts to help us understand how we can communicate the complexity of the human body, not just to experts, but also to general audiences. Then at 10 p.m., Andy, who just welcomed you all, is going to have a behind-the-scenes look, maybe even showing some code from the VR Organ Gallery effort, a way that anyone who has an Oculus can actually come in and explore the 3D atlas in true proportions. Ultimately, also see what tissue blocks exist where, what anatomical structures exist inside of the many, many human reference organs that now exist. At 11 p.m., we have two experts from Indiana University present predictive models of single cells. It's a quite unique line of research, and we're very much looking forward and are very happy that they could join us at such a late time today. Then I think everybody gets a break, because it's midnight. And for midnight, we go back to Amatria, which is a sentient architecture here in Luddy Hall, actually just a few meters away from me right now. And we will have music for human and sentient bodies. And please come back in. And if you are in Bloomington, Indiana, come check out Amatria, say hi to her. We believe deeply that we have to find good ways to live and learn and ultimately exist with artificial environments. Then there's another video session, and these videos come from the Visible Human MOOC. And I think Ezra is posting video links right now into the chat. And in these videos, you will see how the Human Reference Atlas is actually created. You have an overview, and then you have many of the tools and infrastructures that have been developed in the initial 4 years of the HubMap pilot phase presented to you. The MOOC is available online, and so the link will guide you to this website, and you can just log in or create a free IU guest account to enjoy all of these materials for free. It's a 10-hour course developed for general audiences. At 2 a.m., we go over to California because it's a Around the Globe event, and Shankar Subramaniam is going to present on predictive models of the human body. Then we go over to New Zealand and get to see how Peter Hunter is modeling the human body at different levels of resolution. And Peter has been also amazing in bringing his research into museum environments. So please come back for that. Then I have the utmost pleasure to introduce a Chinese team, and that team is really a high-powered team doing amazing work on computational cell atlas assembly. I saw the slides. You can't miss that one. Please do come in at 4 a.m. if you can. Otherwise, watch the recordings of the session. Then at 5 a.m., there are more videos from the Visible Human MOOC, including also videos that explain what we are planning to do in the production phase of HubMap, which just started. And in fact, it continues at 6 a.m. with details on how data comes into existence, how now experimental data is mapped to the Human Reference Atlas, and diverse tutorials, but also the great VHMOOC blooper reel, which you will find rather funny. But I think it's important to have fun when you do work. At 7 a.m., there's a video on the Human Biomolecular Atlas where Jeff Spraggins shows the just beautiful and gorgeous data sets that are now coming into existence, not just within HubMap, but also across many, many efforts. But this is a particular focus on the HubMap data. And this is a rather recent talk for expert audiences, but I think it's also very informative for understanding what happens in HubMap right now. At 8 a.m., we have two experts from Europe joining us, Ali Ertürk at the Helmholtz Institute in Munich, in München, Deutschland, and Peter Lee from the University College London in the UK. You might have seen some of the fantastic data sets that Peter Lee's team is now generating, where entire human bodies and entire human organs can be scanned at highest resolution. So he will present more information on that line of research. At 9 a.m., we have the human vasculature as a latitude and longitude system. You might have seen agree on certain latitude, longitude coordinates, if you wish. But here, this team is starting to use the human vasculature to create a coordinate system, a common coordinate framework for the entire human body. At 10 a.m., there is a panel on Human Reference Atlas ecosystems. It's non-trivial to develop data, code, and training materials that are fair, and to do this across the globe using experts from different disciplines, which have rather different value systems, from different countries, which have very different funding setups. And still, it all should fit together and contribute to the overall whole and common good. And how to do this best is really a topic of this 10 a.m. panel. At 11 a.m., we have Visions for the Future, and there are some brave souls which agreed to project forward how to create this Human Reference Atlas and what we might get to see in the next 5 to 10 years. And so please come back for first comments on how now science catches up to original movies that some of you might have enjoyed in 1966. This is the Fantastic Voyage movie. If you haven't seen it, you might like to check it out. But also using modern infrastructures such as Kaggle competitions to hack the human body. That's the title of the most recent competition we run in close collaboration with the Human Protein Atlas. So please come in, join us for a 24-hour journey. I'm going to give it over to Andy to show you more about what is going on. And before I do this, I would love to thank everybody who was involved in compiling all the materials and keeping us safe on Zoom, and in, of course, tweeting and news iteming around the globe. So Andy, I'm just going to put this here on Dia show, and it's all yours. Thank you all.
Thank you very much, Katy. All right, folks, thank you so much for having me. It's great to be here today. I am going to give you two things. One, I'm going to give you a little bit of a pre-taste of the session tonight at 10 p.m. where we talk about our HRA organ gallery as far as it has been developed so far. But I also want to bring in modern technology that has kind of gotten negative press recently, but that I think can be highly useful for us as we are mapping the human body, and that is virtual reality. I did bring a virtual reality headset here with me. This is the Oculus Quest 2 or the Meta Quest 2. You will see that this particular headset here, if you have ever used one, it is very small, very lightweight, and definitely one of my favorite pieces of VR to work with. So again, my name is Andy. I'm the research lead here in Katy's lab. I want to talk first, when we talk about virtual reality, I want to talk about nomenclature. And so there's a lot of nomenclature floating out there: virtual reality, augmented reality, mixed reality, hybrid reality, but what does it all mean? The funny thing to me is that much of these immersive technologies have been foreshadowed by Hollywood movies. So you can see here a series of screenshots and movie posters out of Hollywood. On the left side here, we see the movie poster of Ready Player One by Steven Spielberg from 2018. In the bottom here, we have Tom Cruise in 2002's Minority Report, an excellent movie that shows him solve crime, like put together timelines via this augmented reality interface. And I have no idea how this would actually work, but it looks extremely cool. And then on the right side, you can see a whole bunch of screenshots from one of my favorite movies, which is The Matrix, which of course is a movie about humanity being trapped inside a machine world and being used as energy sources while their minds see projections of the world as we see it here, which is just an amazing concept. I always joke like it is kind of like going into the Matrix when you put on one of these. And then the only device that you see here that's actually real is on the left side, which this lady here is wearing. This is a HoloLens 2, the HoloLens being an augmented reality device. So to look at this in a little more scientific way, there is the concept of the Reality-Virtuality Continuum by Milgram and Kishino from 1994. So they described that we could understand immersive technologies on a spectrum going from the real environment to a virtual environment. You can see that in the bottom here. So on the left side, this is a picture of a puppy with a runner in a completely real environment. There is no technology, no overlay whatsoever. On the opposite end of the spectrum, we have an entirely virtual environment. This is from a video game called Half-Life Alyx, where you can see everything in the scene is modeled. So everything you see in a virtual reality headset is entirely modeled. But then you have a huge mid-spectrum here, which is referred to here as mixed reality. So here, for example, this is an example with Pokemon Go, where you have this garden, this yard, which is completely real, completely physical, but then you have this virtual augmentation. So you have an augmented overlay. It's augmented reality, mixed reality. And then Milgram and Kishino introduced two terms here, or emphasized two terms here, and only one of those two has survived. So one is the term of augmented reality, which is still, a thing today. Like when you use Snapchat, for example, you can turn on filters. That's like probably one of the most prominent and widespread uses of augmented reality these days. And on the other hand, you have augmented virtuality. This is a term that did not survive. Augmented virtuality, if you've seen that before in the 90s, there were some video games that had virtual backgrounds, but then real actors that were put in there via video. Really funky these days. This technology just did not survive. But this spectrum allows us to understand immersive technologies along a spectrum, and it allows us to describe it allows us to describe these technologies in reference to each other. And then Milgram and Kishino introduced three dimensions. One is the extent of world knowledge, and that is especially interesting for us. So on the left side here, when we have this puppy image here, the world is completely unmodeled, meaning we have no digital information whatsoever about the physical world that is presented to us. Whereas on the other end, the world is completely modeled. You can see that here. So everything that you see in this virtual reality display is completely modeled. That's a very important hallmark of virtual reality, is that everything, every bit of content that the user sees is manipulatable via code. And then, of course, you have this huge area in the middle where the world is partially modeled. So here, for example, for this yard scene, the Pokemon is completely modeled, and we have some basic understanding of the yard. Maybe we have an understanding of the ground and maybe the fence in the background, which we can get via photogrammetry or other technologies. But otherwise, we have limited knowledge about this yard. The second dimension, and this is where, the age of this continuum really shows from the 90s, is the extent of presence metaphor. So on the left side, we have, monoscopic imaging, just kind of like basically bad image quality, based on a monitor. And on the right side, we have real-time imaging, HMD, surrogate travel. Obviously, many of these terms did not survive into our modern times. If you put on an Oculus Quest, you're greeted with a fairly okay resolution. You have stereoscopic view, and nobody really would do monoscopic virtual reality anymore. And then finally, this is again showing the age of this model, is the reproduction fidelity. So on the left side, we have simple wireframes. If you have done 3D modeling before, you're familiar with wireframes, where you essentially just see the vertices and the edges. And the right side, 3D HD TV, real-time high-fidelity 3D animation. Obviously, this is an outdated concept. Essentially, good graphics, bad graphics. So this is an academic view on virtual reality. But what I mean in virtual reality in this talk is exactly what I'm holding up here. A modern-day virtual reality headset with two controllers right here. All right, moving on. Want to talk a little bit more about different types of virtual reality. There has recently been a rise of consumer-grade virtual reality. There are many different options now in terms of price, resolution, field of view, size, and tracking. And there are two important metaphors that I want to show. One here is tethered virtual reality. The other is inside-out tracking virtual reality. So some of you might have used this type of virtual reality before. The HTC Vive or the Valve Index is one of these examples. When you are in virtual reality, and actually going to walk around here real quick. If you are in virtual reality and you wear one of these, the headset has to somehow track where you walk and where you look. And there's different ways of doing that. One way of doing that is to have external tracking stations. So as you can see here on the left side, you can see this person here is using a VR headset, but there are these tracking stations in the room that spray the room with infrared light that allows the headset to track itself. And then here, same for the controllers. And then at the same time, the headset has to be tethered into a computer. And you can see that actually here, this is what I did my dissertation with, is the HTC Vive. Goes into a link box, and then the link box has an HDMI and a USB cable going into a machine, like a PC, and at the same time also this power cable here. Now the Meta Quest 2 is different from that because the headset tracks itself through a bunch of cameras that are added here. This is called inside-out tracking. So this allows us to use this headset without being connected to any kind of high-powered computer whatsoever. Now, for medicine and biology, there has for a long time been this vision of using these these like virtual and augmented technologies to do good for patients. So on the top left, this is a screenshot from one of my favorite TV shows called The Expanse, where we have a victim of a gunshot lying here. And then we have one of the crewmates trying to remove the bullet. And you can see that this is Alex, the pilot here. He holds this kind of like laparoscope-ish kind of tool looking into the body. And then here we have this kind of virtual, this like augmented reality representation of the inside of the patient here, Felsha, who's lying here. Trying to find that bullet. On the right side here, this is very clearly a stock image of what are supposed to be surgeons, I imagine, looking at a 3D model of a patient's brain. The idea here being that, so there's many things of here that are unrealistic with the current status. So one is none of these researchers here is wearing a head-mounted device. This one doesn't wear anything. So they're looking at a 3D model just kind of floating in space, kind of like a hologram in Star Wars, which of course we don't have this technology yet, unfortunately. And this is probably supposed to be the brain of the patient that they're going to do surgery on, which is also something that we don't quite have yet, not at this level of detail. But this is a vision that has been driving the development of virtual and augmented reality for a long time. Now, the reality of using virtual reality for medicine is a little bit different. I would say a little more basic, but still extremely useful. So here on the top left, you can see an app that teaches anatomy students. Anatomy is a very classic application area for virtual and augmented reality because it allows you to show something that is normally kind of hard to get, in this case, a human body, at multiple levels of multiple views. You can see here, you can take the muscles apart. Here you can even take organs out in this kind of, kind of creepy looking environment. And increasingly now there are usages of virtual reality to show histological data. This is from a paper here, a virtual reality for 3D histology. And this figure here explains how the application was being built using Unreal Engine. And beyond that, there are a lot of different use cases for virtual reality also, which you can see here on the right. So for example, for teaching people how to do maintenance, in this case for pilot training, and for operating heavy machinery. This is a really cool example here that I found of someone trying to learn how to use a crane using this kind of a joystick setup together with a virtual reality headset. So one of the really cool things about virtual reality then also is the amount of input that it allows a user to have. So with a typical mouse, as you can see here, I'm actually having one right here, a typical mouse has a left mouse button, a right mouse button, a middle mouse button, or a wheel, and an X and Y position. That is all of the input that you get with a device like this. Now, on the other hand, here on the right side, you can see what all you can do with a virtual reality controller. So you have a variety of buttons that you can click, to a certain degree or completely. You have a joystick, you have a 3D position and rotation. And of course, you have that twice because we are humans, we have two hands. So the amount of inputs that you can give is just staggering and a lot more versatile than what a mouse is. Now, of course, at the same time, there is a reason why the mouse has been around since the 60s, since Doug Engelbart invented the mouse and presented it at the mother of all demos, because it is a very easy to learn input device. But I think that with some training, every one of us can learn how to use one of these virtual reality input devices. So I've showed you a lot about virtual reality, but the question is always, where is the data? How can we show data in these virtual environments? And this is kind of bringing me to personally my research area, which is data visualization in VR. So this is a quote by German computer scientist Daniel Keim from 2001. Visual data exploration seeks to integrate humans in the data exploration process, applying their perceptual abilities. The basic idea is to present the data in some visual form, allowing data analysts to interact with it. So what this quote really speaks to is the symbiosis of computers and humans that is only possible through visualization, because visualization is for humans only. A machine does not need a visualization of a data set in order to understand it, for lack of a better term. So, of course, many formalizations have been made for making, interpreting, and teaching data visualization. And one of them is the Data Visualization Literacy Framework, which you can see right here. This is just a small excerpt from this framework. Everything is written up in this paper that is linked down here. So for example, you can see here different types of color coding, so categorical, sequential, diverging, and cyclic, different types of reference systems, and then different types of visual encodings. And all of these are relevant for virtual reality as well. Of course, there are many perceptual challenges in virtual reality, though. So one is that 2D is typically simplicity, but virtual reality is three-dimensional by nature. And so there are two important issues that come with having a data set in 3D. One is the problem of occlusion. Occlusion is a very important depth cue. If you see something and there is something behind it, for example, you can see the microphone here and then you can see my face. But my face is occluded by the microphone. And because it is occluded, it tells you that I am farther away than the microphone. This is a very important depth cue, but of course it limits what we can see in 3D. And so this kind of got Munchner to quote Ware by saying that we experience the world not in 3 dimensions, but in 2.05 dimensions. We see very well left and right and up and down, but depth is typically occluded. Another problem is foreshortening, which shows a size difference where there should be none. So for example, both of my hands have the same size, arguably. But if this hand is closer to you than this hand, then this one is going to appear longer. This is the foreshortening effect. And so in a data set, of course, if you have data visualization and two dots of the same size, but they're at different distances from a viewer in VR, they're going to appear that they have different values, and that is a problem. Then, of course, the other problem is that 3D has a time and cognitive effort costs. And so we should avoid using 3D as just a purely aesthetic choice. You can see the foreshortening effect here, by the way. These two cubes have the same size, but if I look at it in 3D, this one is going to appear much bigger than this one here in the background. And so to kind of get this back towards the human body, this brings me to the concept of information-rich virtual environments, which is a concept brought forth by Baumann and his co-authors. And so an information-rich virtual environment is a realistic virtual environment, so a VR environment, that is enhanced with the addition of related abstract information. And here you can see kind of an early example of an information-rich virtual environment showing the human throat. So we can see that this is supposed to be a tongue, I assume. And then you can see all of these like overlaid additional abstract pieces of information, such as, this simple label that shows you the name of what this is, the right palatine tonsil. And then it also shows you what I assume is the area. So that does seem pretty large. But this is what all of these related abstract pieces of information are supposed to show. So we can take 3D organs of the human body and augment them with additional information. And so this actually brings me to the organ VR gallery that we were teasing earlier, which is also called the Human Reference Atlas in 3D VR. It is our vision to take the work that we are doing in HubMap and display it to users in virtual reality. So here on the right side, you can see a little bit of just a view of what the application currently looks like. If you're interested in doing a deeper tech dive and meeting the team that is currently working on bringing this vision into fruition, tune in tonight at 10 p.m. So again, the Human Biomolecular Atlas program, or HubMap, and other single-cell mapping efforts have contributed to this by procuring all of these tissue blocks here. And so this organ gallery integrates 3 data types for human tissue. Spatial, meaning where does the tissue come from? Biological structure, meaning what kind of anatomical structures, cell types, and tissue anatomical structures, cell types, and biomarkers belong to it. And then also the specimen and clinical metadata. So what is the donor, age, sex, BMI, etc. Although I'm not going to cover this in this talk. You can look at the currently existing code here on GitHub. This is all, of course, freely available. This is a slightly dated preprint from the very, very first version of the organ gallery, which you can see here on OSF. And again, please tune in tonight at 10 p.m. to learn a little bit more and to also get a live demo of what the application currently looks like. So in conclusion, virtual reality and data visualization gives us superpowers. While there are many challenges for it, specifically perception, the complexity, and also finding the right use cases, and also, getting the correct hardware, I do believe that virtual reality, specifically for building an atlas of the human body, gives us unique powers. So whenever I put on my VR headset and I look at these two human bodies, I feel like I'm having superpowers by being able to look into these bodies and seeing these organs at their true scale rather than just on a two-dimensional screen. And so this allows us to create an information-rich virtual environment in VR and bring this kind of vision that Bauman had to fruition for the Human Reference Atlas, while also integrating the spatial and abstract data into one continuous and immersive environment. I want to briefly acknowledge my team. You will meet all of them tonight at 10 p.m. And then we're going to have a little introduction so that you get to know all of these fine folks. And then here are some references, obviously very important. And I want to end my segment here with a huge invitation. The NIH, together with a series of junior investigators such as myself, are inviting everyone to a junior investigator meeting. This was supposed to happen this year, but we had to postpone it. So because we wanted to have it in person. So this meeting, as you can see here, will conduct several workshops with potential topics ranging from career development, equity in science, to the academic and the industrial world. We are meeting on Roosevelt Island here in New York, here on the East River, in the Cornell Graduate Hotel, and also virtually, but we do encourage participation in person. March 1st to 3rd, 2023, we will put out a website. We are almost done pushing that website out. We are inviting everyone who is a graduate student, a postdoc, a staff scientist, or early career faculty. Please contact me directly. Maybe David, maybe you can put my email address into the chat real quick, please. If you have any questions about this NIH Junior Investigator meeting, please contact me. We are soliciting broad participation. All right, thank you. And with that, I think I'm going to give over to Todd for the trivia, if Todd is ready. I see Katy not. All right. Thanks, everybody, and see you tonight at 10 p.m.